Skyepharma, a biotech company where she served as global project manager for the second drug product candidate. And in 2020, Vanessa joined Skyepharma first as a project manager in a new product introduction department before moving to her current position as Skyepharma Innovation Manager.
Thank you, Aline, for the introduction.
Yeah.
So let's move now to real-life applications.
Third session of this day.
Industrial application, because this morning it was real-life application also, but not necessarily in industry. So I will try with my talk to convince you that it's really important to have some huge investments in CMC in early phase to be able to have a right first-time approach in drug development. So my talk will be divided into 4 sections. First, I will try to give you some few figures about the cost of DP development and also the risk of failure in drug product development along all stages of development. Afterwards, I will move to formulation aspects from preclinical to clinical, and I will give you some case studies with experience here at Skyepharma and with some take-home messages. So first of all, I find this graph very interesting to show you the cost of development, how it has evaluated since year 1963. So you can see that at that time it was only $0.2 billion of cost for a pharma company, and over a period of 30 years it has almost been multiplied by 13, reaching the huge amount of $2.6 billion needed to have a drug product on the market. And what is interesting also to see is among these 2.6 billion that 54% of it is directly out of the pocket, so for the experiments, the development, the subcontractors, the staff, and so on, and 46% is in returns to the investors. And only 20% of the drugs are able to generate enough revenues to cover the R&D cost for a drug. So having seen that, it's not surprising that pharma and biotech companies, under the pressure of the investors, for them the top priority is to get to a proof of concept of safety and efficacy of their active ingredient, rather than working on the best formulation in early phase. But this may be not a good approach to have. 2 other graphs. The first one is regarding the ventilation of those costs on the different development stage, and you can see that the most time-consuming and also costly phase is when you get to phase 3. So necessarily, what will be the priority before moving to phase 3 is to securize the drug product. So this is the really last stage in which we should invest in CMC. Regarding the duration, you can see that all these development stages will last in best case 10 years, in worst case 15 years, and there is this race to market, but after a while you need to invest in CMC before moving to phase 3 to make sure to get the approval and turn your research project into a real drug for the patients. So neglecting early phase formulation would induce multiple risks. First of all, if you have a poor bioavailability of your active ingredients, you will have a low therapeutic effect. You may have also some stability issue. Your active ingredient may degrade over time during stability studies, or you may even generate impurities that may be toxic after a while. You will have some manufacturing issues at scalability, and you will have difficulty to get batch-to-batch reproducibility. Safety concern, if you have off-target delivery, and also toxic impurities. Regarding the regulatory application, regulatory agencies are very eager to have a clear strategy for CMC development and does not like the changes. And they really like to have a really robust data package regarding CMC. And if you have not that, you may face some regulatory rejection for your project. You may face increased development cost. If you have not a good approach, you may have to redo preclinical evaluation, even some clinical study, and maybe it would end to the termination of the drug product if you have not chosen the right formulation. Patient non-compliance. Again, if you don't choose the right drug product, you may don't get the adherence of the patients, and we will have difficulty to interpret your clinical data. The same if you don't control enough your drug product, you will have a huge variability along clinical study between patients, which will give you difficulties to interpret the clinical data. Another aspect that is really important is, as we have seen, the investments are huge for drug product development, and it's very important to securize high value of your project with intellectual property. It can be on the drug substance. We've seen this morning with Julien with the solid state. There are something to do with intellectual property at this stage, but also with the drug product development. If you choose an added value oral technology, in that case for Skyepharma oral, of course, you may get access to intellectual property as well. And another risk is to delay the time to market if you don't choose the good drug product in the end. You may have a delay in your product development and in the sales of the product, of course. So it's always anytime you lose, it's less revenues for you. So it's not that's what you want. So I will summarize in just 4 words: rush now, and you will regret later. So I would strongly recommend to invest CMC earlier than phase 3. That is the real deadline to invest for a successful market access. So in which stages formulation can help and make the difference? It can be already at the animal study, so we have talked about this morning, to avoid discarding some good candidates for toxicity issues, bioavailability, but also in early clinical phases, you need to choose the right oral solid form for phase 1 to avoid insufficient drug exposure, toxicity and side effects, and also for phase 2, when you have an increasing number of patients, you want to have reliable clinical data to be able to interpret them correctly and to have the best efficiency as possible and limit the manufacturability issues because you progress in the drug product development and you want to make sure that you properly control the manufacturing aspect of it. And also, formulation can be of great benefit post-approval to develop an added value medicine with a tailor-made formulation. So I will move on for formulation aspect from preclinical to clinical. So, as mentioned this morning, we are working on a co-marketed offer with Sequence to help discovery people talk to galenic people and make the bridge between these two worlds that are today's separated and increase the successful development. So first, as we have mentioned this morning extensively, it's extremely important to investigate the features of the molecule by itself, and after to also have in-depth characterization of the solid state of this active molecule to make sure that it will be readily usable for a solid form. After, when moving to the mice studies and bigger animal studies that are costly, it's extremely important to properly screen the compatible excipient and make sure you choose a good vehicle for this animal evaluation. And after, when you go to the solid dosage form, you need also to look at different parameters to make sure you choose the good form to ensure a successful phase 1 trial and proof of concept of safety, efficacy, and eventually first sign of efficacy. Yeah. Safety first, but efficacy can be good to check at this point. So here is an overview of the offers that we have developed with Sequence. So it starts really, it covers all the discovery and preclinical field up to the phase 1 clinical trial material. And the first part is mainly addressed by Sequence, working on the API, we have seen that, and developing and screening the best polymorph and the best solid state for the molecule. And after moving also with Sequence on non-clinical formulation to finally end with the best dosage form that will be tested in first patients. So as I mentioned, preclinical formulation is extremely important. Animal PKPD, so the study where you will evaluate the pharmacokinetics and the pharmacodynamics of the molecule are costly, and also the toxicity studies. It can be single dose study or repeated dose study, and it's roughly between 200 to 500 kilo euros. So you need to first do it in a rat, and after to choose another bigger animal to make the demonstration. And roughly 10 to 20% of drug candidates fail at preclinical stage, mainly for the reason that the results show poor pharmacokinetics and toxicity issues at this stage. And knowing, because we have extensively that this morning as well, 70% of new chemical entities are poorly soluble, so BCS class 2 or 4, you understand why it's extremely important to screen a proper vehicle to ensure the success of your preclinical study. So the methodology applied is to profiling the active ingredient, to perform an excipient screening, and to pre-formulate, anticipating also the process that will be used. It can be hot metal extrusion, it can be spray drying, it can be many things, but you need to address that readily from the animal studies. And in that, by doing that, you will increase your bioavailability, maximize the efficacy, and reduce the toxicity. This is an example of formulation that can be already tested in rat and upscale after for human. So this is developed in collaboration with LAGEP. So this is lipidic nanoparticle developed for the oral route. And here, as mentioned by Stephanie, we have already performed an animal proof of concept using abiraterone acetate. So it's an oncology product developed for the treatment of prostate cancer. And this evaluation in rat showed a superiority of nanoencapsulation in terms of systemic delivery. So we observe a 2.5-fold increase in Cmax, only slightly delayed Tmax. So this is a good application because the log P of abiraterone acetate is 5. So as we've seen this morning it's a good candidate to apply to nano-encapsulation. After you have performed your preclinical evaluation, it's time to move to phase 1 and properly choose the oral solid dosage form. So of course in animal you can test already some tablets and some oral form, but you need in that case to choose big animal like dogs, mini pigs, and it's not necessarily very representative of what you can observe in human. So usually you rapidly move to phase 1 with a tailor-made formulation. So first we have seen you need to check the active molecule and the solid state, but you need also to anticipate the active ingredient behavior in the body, to consider the patient population characteristics, also not to forget the therapeutic target and the active ingredient mechanism of action, and to ensure success, to have a well-documented project definitions and objectives. So the first pillar, understand the starting material. Is my molecule soluble? BCS2, BCS4. What is its log P? Should I rely on bioenhancing oral technology that can already tested in preclinic? What is the PSD of my molecule? Should I micronize my API? Is my polymorph stable and reproducible? Is it needle shape? Were there already non-clinical formulations? So sometimes it's important because as CDMO we don't always have the information from the customers, and sometimes there have been some already excipient tested maybe in preclinical, and there is not the bridge with the galenic formulation when you move to clinics, whereas it's really important information that as CDMO, our project manager get when they start a project. Anticipate active ingredient behavior in the body. Does my active ingredient rely on active transporters to reach the blood? Where are located the active transporter in the GI tract? How food will impact my drug? Is my molecule metabolized? Does it undergo metabolization through cytochrome 3A4? Is my API substrate of PGP? What may happen if I do have off-target delivery? This is important also to anticipate that, to avoid any toxic effect when we move to clinics. Does my API have any detrimental effect on the microbiota? Indeed, drug microbiomes interaction. It may be a growing concern in modern therapeutics. As you may know, gut microbiota plays a critical role in numerous diseases. Neurodegenerative, metabolic, autoimmune diseases, we say that it's a second brain. So everything is interplayed with the molecules that are excreted by the bacteria we do have in the colon. And I wouldn't be surprised that in the near future, the authorities would require to study the interaction between new medications and the gut microbiota to ensure that you have the good approach in terms of formulation. It's what we call pharmacomicrobiomics. So it's in this study you may evaluate the interaction of the drug on the microbiota. Will it destroy a certain species of microbes? Will it induce different heterogeneity of the microbes you have in the colon? And inversely, is your drug metabolized by the bacteria that are present in the gut or in the colon? Because it will have an impact on the pharmacokinetics and in the end an impact on the therapeutic effect of your drug. So as you may know, in the body we have a gradient of bacterial density, and definitely the colon is where the microbiota is the most representative. So that's why also at Skyepharma we have developed mucotabs So this platform has been developed to have mucoadhesive tablets that target different regions of the GI tract, but we have this specific one that is designed to have a retention in the upper GI tract, and it could be used to prevent the microbial interactions of your drug with the microbes. So it's a combination of mucoadhesion and pH selection, and so you can target different regions, as I mentioned previously, so stomach, duodenum, intestine, and we are currently under development, but we have a ready-to-use formulation, and we demonstrated that we can reach up to 24-hour active ingredient release in the upper GI tract. So in that case, we would avoid the colon, which may be a danger zone when it deals with your active ingredient. So the third pillar to consider is the patient population characteristics. Is it a geriatric or pediatric patient population? So this one is easy. Is the population gender specific? Indeed, you may not know that women have a longer I have yeah, women, it takes more time for the women. Yes. Flush. Yeah, flush. Sorry. I will find the. Huh? Yeah. So women, it gets longer for them to excrete the drug. And there has been this case with extended release theophylline tablets in which it was observed in women a longer and greater extent of exposure of the drug. So it was theophylline was used to treat respiratory conditions like asthma, and they observed some side effects using this drug in humans. And they were experiencing palpitation and nausea because they were exposed with the same tablet. They were greater exposed, and it induced some side effects. So you need also to think if your population is immunocompromised. Do they have also specific microbiota? Do they present normal transit time? Do they present different pH values along the GI tract? And is the target population from exotic geographic region, for instance? It may drive also the strategy you put in place regarding stability studies. And the last one, you need also to fine-tune the DP product to the therapeutic target. Where is located my therapeutic target? What is the mechanism of action of the molecule? Do I need to ensure a sustained exposure to saturate the site, or is a sharp but intense exposure preferable? Do I need to target the stomach, the colon, a specific region? And also, you may also want to combine some active ingredients. Is there any priming of the target needed with another drug that would be requested to activate your active ingredient? And also, you can combine ingredients. For example, if you want to cure the side effect of the first one with a delayed approach, so you can have an active ingredient which has side effect, and you want to counterbalance this side effect with another API that would be released later on in the same tablet. And last but not least, the importance of the QTPP when moving to phase 1. So pharma and biotech companies may have already in place target product profile that drives the overall development strategy. So it's a document that is strategic for the company, and it's used across different functional teams. So it's very high-level document that defines the clinical strategy, the therapeutic goal, and the commercial goals. but it's also along with this document to associate a quality target profile. This is also a strategic document that needs to be shared with the CDMO, and working together will give a better result in terms of drug product development. So the quality target profile is more dedicated to describe all the detail and the target needed to have a good quality of the final DP product, and clearly details the attributes, the critical quality attributes that are necessary to ensure the safety and the product performance in the targeted patient population. So I will now move to some case studies that we have experienced at Skyepharma. So the first one is for an first-in-class active ingredient for the treatment of Crohn's disease. So it was an hydrophilic active ingredient, gut restricted, meaning that it was poorly permeable and it was not what was wanted, since the therapeutic target is in the GI tract. So for the phase 1 and phase 2A formulation, an immediate release monolayer tablet was used, and the drug product development strategy was, as usual, fast clinical launch, minimal investment in CMC, simple and cheap formulation. And this customer came in phase 2 with CMC investment because he wanted to have, at the end of phase 2, so to initiate the phase 2 trial, he wanted to develop a new oral solid dosage form to explore a better outcome of the treatment efficacy. And we have mentioned that previously. So he relied on GastroPlus simulation. So the data entry in this simulation was a 300 milligram immediate release twice a day regimen treatment. And it was compared head-to-head with a 100 milligram gastro-retentive solution twice a day. And the outcome was that the target product profile needed in terms of dissolution was a slow release over a period of 8 to 10 hours in the stomach to result in a maximal exposure and a greater efficiency at a lower dosage. So for this customer, we proposed our SOCTEC technology. So SOCTEC is a self-oriented capsule flotating in the stomach. So it remains vertically thanks to an empty capsule and a ballast that are in the interior of the capsule. After that, so these are the flotating elements. And after that, we perform polycoating. So the first one is enteric coating, also to ensure the integrity of the capsule in the stomach environment. The first coating after is the active ingredient, followed by another coating, which is a mix of sustained release polymers, a mix of hydrophilic and hydrophobic polymers to end up with a sustained release profile of the active ingredient. And here we have a representation of the capsule along the different steps of coating. So here on the left, you have the result of a clinical study we made with placebo SOCTEC, in which we demonstrated that after 9 hours, we still have a gastric retention of the SOCTEC technology in the stomach of 75% of the subject. And on the right, you have the dissolution profile of the active ingredient that was coated on the capsules of SOCTEC, and we managed to reach the target profile of the customer with a delivery of 80% of the active ingredient in 10 hours. Another case study is regarding an investment occurring in phase 3 clinical trial. So the phase 1, phase 2 formulation were immediate release monolayer tablets with an active ingredient that needed a pH modifying agent to promote the systemic delivery and result in a fast Cmax and Tmax in the blood. So the drug product development strategy again was fast clinical launch, minimal investment, but there are stability constraints with the product because the drug product was stable only at 2-8 degrees and had a short shelf life. So before moving to phase 3, there was a CMC investment to investigate better the formulation, and obviously it was an incompatibility between the active ingredient and the functional excipient that was observed and resulted in this reduced stability of the drug product. So what we did propose for this customer is our geometric multi-layer tablets. So the approach was to introduce the active ingredient in a first layer, immediate release layer, and to have this pH modifying agent in the bottom layer, and to separate the API from the functional excipient with a protective layer. So the formulation benefits in that case is that improve the DP stability. So you switch from cold to room temperature stability. It provides longer shelf life. It improves the supply chain for clinics, but after also on the commercial phase. And it brings better comfort for patients because it's a simplest storage. So the impacts for this latter case of the late CMC investment on DP just before phase 3. We have mentioned that it is the real deadline to make the CMC investment before moving to phase 3. But what are the impacts on so late investment? The first one is financial. First of all, you need to redevelop all the analytical DP methods. You need also to collect new stability data, and you need to modify all your regulatory DP section for your regulatory dossier for the phase 3 studies. You have also some industrial impact because you have decreased knowledge on the process before the process validation, and you have also manufacturing risk at commercial scale because you have less historical data. And there are also regulatory and timelines impact because you experience more than 6 months delay launch of the phase 3. You compromise the QPD approach that is required for FDA submission. You may raise CMC question for the approval, and you have also a delayed market launch and commercial scale. So that is why it's preferable to invest in early phase. The case study number 3 is for a post-approval project. So it's regarding Xeljanz, which was developed by Pfizer. So it's a JAK inhibitor, tofacitinib. First-in-class molecule developed as a monolayer immediate release coated tablet for the treatment of ulcerative colitis and other inflammation conditions. So it received the approval in 2018, and in 2021 it received a warning letter from the FDA because actually there was off-target delivery of the molecule leading to serious adverse events. Among them there was heart-related events, cancer, blood clots, and even death. So this had some serious consequences for Pfizer, of course. First of all, financial impact, because they need to implement some additional post-marketing studies. They observed a decline in the sales, of course. They needed to multiply the communication campaigns to see the doctors because they experience and reduced trust in the drug, and to revise also secondary packaging to include all the risks mentioned and so on. But there was also reputational damage because it is very harmful for the company's reputation, and you may experience also a loss of confidence in the marketed drug. And legal impact, because of these side effects, you can have some potential litigation and lawsuits related to the serious adverse events that were observed in patients. So for that kind of product, there is an opportunity, of course, of added value medicine. So here, a good technology that could be applied is GeoClock. So it's a delayed release technology that is already marketed at Skyepharma under the brand name of Lodotra or Rayos. So we do have a product for rheumatoid arthritis with using the active ingredient prednisone. So here we could do the same, have the JAK inhibitor in the core tablet, and so it's a tab-in-tab. The outer shell is inactive shell and responsible for the delayed release effect. And we could divert this technology to target the colon region by adjusting a 6-hour lag time, meaning that you would open the shell and deliver the active ingredient 6 hours after the uptake of the pill, which would nearly target the entry of the colon. So how does it work? We are able here at Skyepharma industrially to control the way of we tablet the core and the shell to adjust the lag time between 2 and 8 hours. And how does it work? Along the GI tract, along the waiting with the biological fluid, there is an initial absorption of the liquid which initiates a breakage of the shell, and which in the end separates the shell and release the core tablet, which release in the end the active ingredient in an immediate release manner. So for this kind of added value medicine for a post-marketed drug, what is very interesting regulatory pathway to apply is the 505(b)(2). So it's a regulatory FDA pathway for the US market. It's extremely interesting because contrary to the generic here, you need to demonstrate that you have a really added value of your technology as compared to the reference drug. So you need to provide supportive data to show that either you have a different profile or an added value compared to the original molecule. And it's reduced development cost and time compared to a full new drug applications, and also it's regarding the commercial price, it's very advantageous because there is no strict regulation for pricing, on the contrary to Europe, where it's begin to change the mind regarding the added value medicine, but we are not so well advanced as for the US, where this regulatory pathway exists. So for this kind of project, from scratch, you can have roughly this timeline. So you begin with prototypes along with analytical development. You can include in such a development after a year some pilot PK study to investigate one or different prototype and make sure you have the desired PK profile. And after to move to the pivotal study to be able to have your 505(b)(2) application in 24 months. So to conclude on the benefits of early investment in formulation, so it's very beneficial to have this early investment for improved efficacy toxicity ratio, for the patient compliance, to enhance the stability and the manufacturability, to reduce the cost and to avoid to redo some work that could have been avoided if there have been a good thinking from the beginning, to ensure regulatory success and also to have a strong IP protection, which in the end will guarantee that all the costs will be covered by the commercial sales. So thank you for your attention.
Perfect timing too. Is there some questions for Vanessa? Perhaps what you mentioned during on one of your slides, QBD approach. What would be your recommendation to introduce QBD at what stage, for which topics, etc.?
So quality by design can actually be done at every stage of drug product development. So yes, at Skyepharma, this is something that we standardly conduct with a systematic approach, starting from a risk assessment of the pharmaceutical operations, defining the most critical operation along the process with FMEA, so failure mode effect analysis, and after running the QBD through a design of experiment to be able to properly define the design space of the process. So a QBD can be run on the excipient, on the drug substance, on process parameters. So it will depend on the outcomes of the risk assessment. This is really the outcome of the risk assessment that will drive where you should invest in quality by design DOE approach. And it's very helpful also to have this kind of data package before moving to validation of the process, because you know what are the limits of your process to specify as a specification during the process.
Other questions for Vanessa? Okay, thank you, Vanessa. And so, last speaker for today is Quentin, and not Bruno, but from Medelfarm. So Quentin Boulet is marketing director at Medelfarm, chemical engineer as a background, and he joined Medelfarm 5 years ago. The title of Quentin's presentation is Still One Compaction Simulators, a tool for accelerated tablet development and efficient production support.
So, good afternoon, everybody. So, I'm not Bruno. For those who know him, or if you look at the program, so I'm just a younger version of him with more hair. So, today I will cover the topic of the Style 1, how it can be used in tablet development, but also later on in the process when your tablet is filed and in production. But before we get started, I will just introduce quickly Medelfarm and Korsch, what is a compaction simulator and how we can use it at a different stage into the development and in troubleshooting and production support. So since 2 years, we are part of the Korsch group, so it's a family business in Berlin, specialized in tableting technology. So from R&D to production, so high volume, small volumes, pilot plants. we cover all the needs for tableting technology. So you are welcome to visit the Berlin site if you have a project. So we are part of the leaders when it comes to tablets compression machine. We have many patents. We have a global presence. So we cover all the markets. We have a lot of testing facilities as well. So if you need just punctual support or consulting, or you have facing issues, we can help you. And Medelfarm, inside the core group, so we are based 30 minutes away, close to Lyon. And we are very specialized into the compaction simulation business. So our core business is really to develop, manufacture the Style 1 Evo and the Style 1 Nano compaction simulator. So those are the two machines that you see here. And we also have an application lab where we perform training, testing, consulting, and of course it's part of they are our first customers as well to develop new functionalities. So we have R&D product range. Today I will focus on the Style 1 Evo, but we have eccentric presses, we have small rotary press for development. We can also offer containment application. I'm mentioning this here because of the new zone MMPP where we have a Style 1 Evo Drycon. So those are two different concepts, but the idea is really to protect the operator when you are handling potent API. So of course, different technologies, different pricing, but we can adapt. And Korsch, so as I said, from pilot production, couple hundred thousand tablets per hour, to big volumes, million tablets per hour. So we can really manufacture patches, to manufacture patches in the pharmaceutical industry, but not only cosmetic, industrial application, batteries, many different applications. So what is a compaction simulator? So I think some of you are already familiar with this technology, for some of you it will be new, but I'm sure you all know what is a rotary tablet press. So you have a turret with many punches, many stations, and the goal of a compaction simulator will be to replicate the punch movement like if you were on a rotary tablet press. If you take the rotary tablet press, so the punches moves on the rotary, the turret, but at the end of the day, they move only vertically. So they will go through the pre-compression roller, relaxation, main compression, and then ejection. So this is what you see here. We will move the punches the same way so that they will match the displacement like if they were on the production equipment. And in order to do that, we need only the geometry of the rotary tablet press. So the roller dimensions, their position on the turret, the size of the turret, type of punches, and so on, to build with our algorithm a profile, and then this is really where the know-how of Medelfarm is in the motion, the way we will move the punches to have the very high acceleration, the very high punch velocity to match the same velocity as on the production equipment. So here, it's just a small video showing a compression at low speed. So it's on the production equipment, so a Korsch mid-size equipment at 10 RPM. So you will not produce a significant amount of tablets per hour. So I put, if you look at the lower punch, it moves quite slowly. But then on the right side, it's at very high speed. So here, I will play it again, but I'm sure you have difficulties to watch the movement, the last movement of the punch. And this is what is important. If you perform your development on a small rotary press or on an eccentric press, not using the high velocity. If you are using material that are sensitive to speed in your formulation, you are maybe at risk for tablet defects. When you scale up, you might face some challenges. So it's important in R&D to foresee these tablet defects, to understand how your powder will behave under the production conditions. So how does it work? So we have the punches are instrumented, so we measure the displacement and the force in real time. So for each given moment, we know the position of the punch and we know the force that is applied to the powder bed. This helps us to first drive the tablet press very precisely, but also to get the information about the compression events, the ejection events, and to understand how your powder is behaving. We can do that for single layer, bi-layer, 5-layer tablet. We can also do that for tab-in-tab, so very diverse applications. And we have additional optional instrumentation that we can add to understand what will be the radial pressure that you will get on your die when you eject the tablet. What is the force that you will be required to detach your tablet from the lower punch? You don't want to have high forces because it might create some picking, sticking, or just your tablets will fly in the tablet press. And then the idea would be to correlate, of course, your formulation, your process parameters, the compression data with your external measurements. So the hardness, all the physical parameters of your tablet press, but also friability, dissolution, stability. You can look at many metrics, and this will help you to drive your decision making in early stage or later on during the scale-up process. Just waiting for it. So again, Style 1 Evo equals to R&D at production speed. So for single layer, multi-layer, tab-in-tab, fully instrumented, it comes with the software. You can do the data analysis, build your graphs. Quickly you can get the information and go back to your desk with your brain and just adapt your formulation or the process parameters to meet your targets. So the Style 1 Evo can be used at different stages of the life cycle of a tablet. So very early to understand each individual components. Okay, how do my API behave? Is it plastic, elastic, brittle? My excipients as well, to maybe compare different sources of excipients, and then to combine them and to understand, okay, if I try this ratio of binder, this lubricant, and so on, what will be the impact on my tablet properties later on? But not only the formulation, also the process. What will be the impact of my binder ratio in wet granulation? What will be the effect of milling conditions, and so on. And then, since you are doing your R&D in the production environment, at least for the compression, you will be able to scale up much easily because you will have a starting point, what will be the process parameters to be used on your specific production equipment. So your Fete, your Korch, your Killian, it's not because you are Korch that you can simulate all machines. And then troubleshooting. You're in production, high volume, but you have a lot of capping, or you are experiencing sticking. We can replicate this issue by taking a sample from manufacturing. We can take the tooling from manufacturing, replicate the issue on the Style 1, and then try to solve it, and then to re-implement the new process parameters. Because you don't want to touch your formula. You can only touch a few things in the process. So now I will cover a few examples. Those are basic examples, but I'm sure it will give you ideas on how to use such equipment in your daily life. But to understand your API, your excipient, we have many tools. So we can look at the energies to understand is plastic, elastic. We can look at the elastic recovery, the AKL analysis as well. Is it brittle? Is it plastic elastic? We have additional measurements, like the takeoff force, I mentioned it. We can look at the ejection stress. It's interesting to know the maximum ejection force, but sometimes to look at the signal itself, it will give you the information, is it sticking or just flat? So not only the value is important, but also the shape. And more recently, we developed an instrumented die with we have the ability to change the temperature of the die. So very useful if you want to look at how your API will behave at 20 degrees, at 30 degrees, 40 degrees, because we know that sometimes in the production environment, especially on the production tablet press, the temperature can rise. So we developed these new things to understand, okay, is your API changing in its form? Is it will really stick, or these things? So here we have made an experiment with 3 different excipients, different manufacturing conditions. 2 are crystallized. One is spray-dried, is mannitol. And nothing surprising here, but the spray-dry mannitol showcase more hardness. So it's important to know how each individual component behave, so that you can make the right selection to build your formulation. Because if you are having maybe an excipient, an API that doesn't compress well, which is mostly the case, maybe you will take something that will bring more cohesion into your formula. And here you see as well that it varies with the particle size distribution. So the smaller the particle size, the more hardness we have with this mannitol, which is you have more surface interaction, more ability of the particles to bind between each other. So I introduced the radial pressure. So every time you have a vertical constraint, so the 2 punches compressing the powder, you have an horizontal response. And of course, during the compression, this is the case for all the powders. But once you retract the punches, it's interesting to look at if there is some residual pressure on the die. Because if you are experiencing too much residual pressure towards the die, you are at risk for tablet infectious capping, lamination. And I'm going to show you a very extreme case from a customer in Japan, where the tablet was exploding because of too much pressure at ejection on the die. This is an extreme case. Formulation development, so look at different mix with your API. Here we have in purple a majority of MCC, which is known to be speed sensitive. So the darker purple is at low compression speed. Here it's at high compression speed, and you see there is a loss of hardness. We could repeat this maybe with other metrics dissolution, but we have done it with the disintegration, and you experience the same, which is not the case with the majority of DCP because it's brittle, so less sensitive to speed. same idea. You have different lubricants. You want to evaluate their performances. Maybe we know that mostly people are using magnesium ceriate, but more and more naturally developed lubricants. So it's interesting to look at their efficiency, what level we should put in the formula. But we all know that the more lubricant you put, the more you are at risk for loss of hardness, impact of on disintegration, and so on. So it's always to find the right balance between, okay, I will reduce my ejection stress, I will facilitate my flow, but I don't want to compromise my tablet's properties. So the style one would definitely help you to find the good compromise to meet your target quality attributes. And this is a study that was made by Skyepharma, I think 5 or 7 years ago, on a 6-batch feasibility study. The calculation was made how much material would be needed on a rotary press to perform this 6-batch study. How long will it take? And in comparison with the style one, so quite huge difference. So a gain in material cost, waste, but also on time, because much less installation time, much less cleaning time. So you gain a significant amount of material and time, so cost. So now the scale up. So that was a customer that came to Medelfarm in our science lab. The idea was to transfer one of their products to a Porsche machine. They were using another machine, and they invested in the Porsche production equipment. And they wanted to know what will be the design space, what range of force were acceptable to meet their target attributes. So they wanted hardness above 120 Newton, disintegration time below 50 Newtons, nothing crazy, but thickness. Just to explain you the philosophy, so we're able to plot very easily with just a couple hundred of grams a graph with the hardness versus the compression force. So to determine, okay, I need to be above 16 kilonewton to reach my hardness. We did the same job for the disintegration, same job for the thickness, and we send the sample back to the customer to do the dissolution, because we cannot do that in-house. But at the end of the day, we're able to tell the customer, okay, you should work in this range of force on your production equipment, and you will get your tablets the way you want them. And they actually transfer to their Porsche X3, and it worked. So the idea is really to give you a starting point. You should work in these process parameters. Of course, there is always fine-tuning, because we are not mimicking the flowability of the powder, so we cannot predict how it will flow on the production equipment. So sometimes you have to reduce the speed of the production equipment for that, but at least for the compression event, we work in a safe environment. Troubleshooting, so capping, very common tablet defect. Many reasons for that, many causes. It can be too much air entrapped, too much elastic recovery, non-uniform densification. So you see here, that was a numerical simulation from the University of Bordeaux, showing that there is a high stress point here between the die wall and the cup that explain why the tablet cracked. And here is another example on a very old Medelfarm machine at very slow motion to inverted capping. So you see the cup detaching. So thank you, Vincent Mazel, for this video. So here, this case study is not correlated to the numerical simulation of the video you've just seen. It's something, a customer that again came to us facing capping issues. So we asked for a sample of their powder, the tooling. The first thing is to replicate the issue. So we were able to do that on the Style 1 Evo. So you see a drop here, and it was exactly at that time that we experienced the capping. So we tried many solutions to overcome this, but the first one, that is the easiest one, is okay, let's add some pre-compression. And you see the red curve, we were able to get tablets without defects just by adding 10% pre-compression. But on top of this, we were able to increase the hardness compared to with no pre-compression. So the idea was to go a bit further and to say, okay, why not increasing the compression speed? Because if the customer can gain maybe 1, 2, 3% more tablets on their production equipment, I'm sure the production manager will be happy with that, with the productivity, the increase in productivity. So that's what we did. And we showed that with an increase in speed, we were able to reach the same target just to avoid the drop and the capping. And that was a solution that we brought to the customer. And that's a way to use the Style 1. So we are speaking about capping here, can be lamination, the sticking, loss of hardness because of maybe over-replication. We can look at many tablet defects to troubleshoot the production issues. So it's just a summary of what you can do, a part of what you can do with the Style 1 compaction simulator. So many applications from looking at different sources of excipient to back up your supply, formulation screening. We can even produce small batches. We have a module for that to build like maybe your phase 1 clinical. We have some customers doing this. We have even customers using it for commercial batches, for oncology products, very small batches, or process optimization. You are transferring your production from one tablet press to another. The Style 1 can be a useful tool for this to predict what will be your new process parameters on your new equipment. But you can not only limit to the compression, you can look also at the upstream processes, you can try to optimize that and what will be the impact in production for the compression. And I'll be happy to take questions. I can take a lot of questions because it's warm here.
So questions for Quentin? So perhaps one on my side, so you spoke a lot about compression, so tablet compression. What about roller compaction? Because we use also Style 1 for roller compaction.
Yes, true. Yeah, that's true. I focused really on the compression. One other application is the simulation of the roller compaction process. So for that, we will mimic we will simulate the force. We'll simulate how the powder goes through the rollers in the compaction process and what force is applied. So we have specific compression profile for that. We can mimic a boiler compaction, roller compactor, spot vector, and so on, many different brands. And the idea is to look at maybe different solid fraction, to target different solid fraction, mill that at the external phase, and see, okay, did you increase your flowability? That was the goal. Did you improve your flowability, but did you lose too much of the compaction so that you will not be able to form good tablets after? So there is always the good balance, and we can use the Stalwell Niveau for that. We have many customers using it, many science lab customers as well, many trials.
Thank you. Yeah.
Yes. So Stalwell Niveau, definitely you can do everything. With the Stalwell Nano, it's more, first it has been designed only for single layer application, and it's more for the early phase. So looking at how your excipient behave, looking at different formulation blend, but it has less scale-up capability. So because of the compression speed, which is a bit limited, you will not be able to simulate a production equipment at very high speed. So it's really for the early phase, the development of new excipients, looking at the first formulation screening. But when you really want to look for scale-up, the Stalwell Niveau is the product.
One question about your tablet form, because you show only examples with the round tablet. Can you also make by the same with the same equipment or sticks tablets or other form?
We can do many different shapes. It can be oblong, capsule shapes, with a secability bar.
Breakable.
Yeah. It all depends on the tooling you use. So here, in many cases, for the characterization or the first work, you do with the flat round 1128 because it's easier for the because it corresponds to 1 square centimeter. But then, of course, we can do with the real punch shape with all the standards, Euro, PET, TSA, and so on.
And well, how do you eliminate that? So the powder is coming from what size?
From sorry,
because you press it up. So the powder, where is coming from?
So we have feeders. We have feeders, so we can install several feeders if you are doing multi-layer. But so if you look here, we have one feeder installed. So the powder comes from this feeder that is moving on top of the die. The powder flows into the die, the powder retracts, and then we compress. And we have different type of feeders. It can be gravity, can be with different paddles, or you can fill by hand. If you have 1 gram of material, you fill by hand, make your tablet. At least you have your first feeling of how your tablet will be.
Yeah. And the flow speed is always the same. How do you control that? So how do you verify that the amount of powders is always the same in your tablet?
Flowability, great question. It's a complex thing because here it's a compaction simulator, so we are really simulating the compression event and the ejection. The goal of the feeder here and the different type of feeder that we have is really to try to make sure we fill the die consistently to get reliable data. If there is if you are trying to fill the die with a gravity feeder with a product that doesn't flow at all, you better do it by hand.
Yeah.
But it will be the case also on the production equipment. So we try to bring solutions for the filling with different solutions, but there is no one solution.
Yeah, but normally the fluidity, the fluidity of the powder is quite dependent on the excipients we put in it. No? I think, well, to my knowledge.
Yeah.
In that case, it will be a formulation issue to solve by formulation, yeah.
Dry granulation, maybe, but it's
Because at Skyepharma we experience trouble with the feeding system of the style one, needing to reducing the speeding speed, even setting at almost 0, 1, to be able to prevent from either over lubrification or separation of excipient from the API in the feeder prior it goes in.
But if you are close to 0, do you have a gravity feeder? Because maybe that's the best solution.
Yes.